Hello everyone. Today we're going to continue our discussion about the discovery of the structure of DNA with this relatively messy uh, PDF here. Last time we talked about how they discovered that the DNA, the DNA, this deoxyribonucleic acid, seemed to be related to inheritance. They saw that both through the inheritance of traits such as infection from viruses. So they used radioactive viruses in the Hershey-Chase experiment to show that the radiation that was placed on the DNA would be inherited by the infected bacteria. And they also did the other experiment, the Oswald Avery experiment, where they also showed that if you, as long as you did not destroy the DNA, a bacteria could inherit a new trait. And so this showed that DNA was important. So Erwin Chargaff was working on DNA in the 1940s and trying to figure out some interesting ideas about it. And one of the things that he discovered was that the 4 different bases he would find in DNA, and different 4 bases that he would find in RNA, were always found in a specific ratio. So I do want to talk about these bases for a quick second, so I'm just going to do a little bit of racing here. So there are 5 different bases that make up the nucleic acids in your body. In the case of DNA, DNA is made up of the 4 amino acids cytosine, thymine, guanine, and adenine. So these are all in the family of the DNA molecule. So if you have a DNA molecule, it'll be made of those 4 chemicals. Those 4 chemicals come from 2 different groups of organic compounds: the pyrimidines, which are the smaller chemical, and the purines. These are both nucleic acids, and these are actually nitrogenous bases. Sorry, these are bases. So as per if you're taking a chemistry course, for example, these ammonia groups are basic. So they react with water and make the water's pH rise. And so these basic compounds, there are these 5 of them. 4 of them that make up DNA: cytosine, thymine, guanine, adenine. So just to keep straight, DNA is made of cytosine, thymine, guanine, and adenine. If you've ever seen the movie Gattaca, the title of Gattaca is Gatta, and that's a reference to this formula or this formation. Now, in RNA, the other type of nucleic acid that's in your body, we'll get into the specifics of what's the difference between these eventually. There is cytosine, uracil, guanine, and adenine. And so in this case, there are 4 again, but they are slightly different. So both cytosine is found in both, guanine is found in both, adenine is found in both, but uracil is found in place of thymine in the RNA. So RNA is made of cytosine, uracil, guanine, adenine. DNA is cytosine, thymine, guanine, adenine. Maybe we should have put these in alphabetical order. Maybe that would have made more sense. And then you would have your A, C, and hey, why not? Why not make it true alphabetical order, right? And put the G's in there, and then it'll be like that. Okay, so ACGT, ACGU, so adenine, cytosine, guanine, uracil. Okay, so the interesting thing that he found was when he broke down DNA particularly, he would always find an interesting amount, an equal amount of adenine and thymine. So there was always an equal amount of adenine was always equal to the amount of thymine. So this is all supposed to be read as one line here. So adenine was always equal to the amount of thymine, and cytosine was always equal to the amount of guanine. This became known as Chargaff's rule. He could take it further and say that if you added the amount of adenine to the amount of cytosine, then you would get you would be equal to the amount of thymine plus the amount of guanine. Or if you added all four together, you could find 100% of all the bases that are found in that DNA molecule. So nucleic acids turn out to be polymers, and the nucleotide that makes up this polymer, or what is also known as a monomer, so a polymer is made of many monomers, happens to be carrying one of these nitrogenous bases. So here is the nucleotide over here on the right, and the nucleotide has three parts. It has a phosphate group, a sugar, and a nitrogenous base, and these are the building blocks for a DNA molecule. That DNA molecule is going to be made up of these phosphate groups, these sugars, and then one of the four nitrogenous bases. If this is a DNA nucleotide, then we are looking for, up here, a adenine, cytosine, guanine, or thymine. If this is an RNA nucleotide, it turns out that it's very, very similar. It's going to have a phosphate, a sugar, and a nitrogenous base, but the nitrogenous base is going to be either adenine, cytosine, guanine, or uracil, and the sugar is going to be different. It will be ribose sugar. So each nucleotide has three parts: an acidic chemical made of phosphorus and oxygen called a phosphate group, a sugar, a molecule. This is made of 5 different carbons that comes into play later, and 5 different carbon elements. So let's not measure them out. 1, 2, 3, 4, and 5 carbons there. Maybe I should have used red. So that's your 5 carbon sugar. Sometimes people use a shorthand of 5C. So I'm actually going to label them the proper way. 1, 2, 3, 4, and 5. So it has these 5 carbons. And that sugar can come in 2 types. It can come in ribose, which is found in RNA, or it can come in as deoxyribose, which is the D in DNA. And then it has one of the 5 possible bases, where you have the adenine, thymine, cytosine, and guanine for DNA, or you have the RNA, which is the other one: adenine, cytosine, guanine, and uracil. And so that is called a nucleotide. And these nucleotides, they discover the nucleotides separate from identifying how everything fits together. They understand that the DNA molecule is made of phosphorus. They understand the DNA molecule is made of sugars, deoxyribose sugar. They understand that the DNA molecule has these 4 nitrogenous bases. What they don't understand is how all of these things fit together to make a structure that can store information important to inheritance. That was still being figured out at this time. So I'm going to just show you some examples of how we can use Chargaff's rule to sort of determine some questions. I'm actually going to do these together with you. So why not I'm just going to delete it all. Hopefully that just works out. Okay, so when you're doing a Chargaff rule question, you have to take into account that as much adenine as you have is an equal amount of thymine, as much cytosine as you have is an equal amount of guanine, and if you add up the cytosine and an adenine, or add up the adenine and guanine, you'll get the same amount as you would get for the thymine and the other one. And if you add them all up together, then it comes up to 100%. So let's look at a question here. A chromosome is discovered to be 32% adenine. So 32% is A. How much guanine, cytosine, and thymine does that mean? So the first thing you want to do is use that first equation, that adenine is equal to the amount of thymine. So that means you have 32% of the thymine. Now, you know that adenine plus cytosine has to be equal to thymine plus guanine. And so you can actually predict here that this has to be essentially 50%, right? Because both of these add up together. They both add together to form 100%. So right now we have 32 plus something plus C and 32 plus G. And we know that each of these sides has to be half of the DNA molecule. So how much is C? Well, C must be 18% and G must be 18%. So let's just do a double check. We can go 32 plus 32 plus 18 plus 18. This is 36 and this here is 64. So together that's your 100%. So that is correct. So that's our final answer. That's all of our DNA components. So a chromosome is 22% adenine and thymine. How much of each of the 4 bases make up the molecule? So now we have 22% is equal to the adenine and thymine total, as that's how I'm reading it. A chromosome is 22% adenine and thymine. How much of each of the 4 bases make up the molecule? So adenine is always equal to thymine, so that means that we would divide this by 2 in order to find the total for adenine. So that means adenine is only 11% and the thymine is only 11% as well. And then if you know that, then you can use the same rules we used before. You could also do some trickery like this. How about we use this A plus C plus T plus G has to equal 100. We put in our A and T, which is 22%, plus C and plus G is equal to 100. So that means that C plus G is equal to 78%, right? Yes. And so then if we know that C and G are equal, then that means that we can divide by 2 to get C or G. So C would be 78 divided by 2, which is 78 divided by 2, 39. So C is 39%, and G is also 39%. Okay. And then Steve suggests that if a chromosome is 30% adenine, it must be 15% guanine. Has he made a mistake? So let's see if we can figure this trick out. So here we know that A is equal to T. So if A is equal to 30%, then T is equal to 30%. So that means that that's already 60% of our DNA. And we know that G and C have to be the same value. And then together, they have to add up to the 40%, right? Because this has to be 100. So that means that C and G have to be 20% each. And so we would say that Steve is incorrect. Correct. C and G equal 20%. Okay, and I think we're going to cut it there, and we'll come back and talk about structured DNA in the following lesson. Have yourselves a great day.